Hello again. It's my pleasure to welcome you back to this channel. I remain Dr. Rehan Falari, and in today's session, I'm thrilled to let you know that we'll be exploring one of biology's most fundamental processes, and that's the genetic control of fertilization. Of course, fertilization is far more than just a chance of meeting between the sperm and the egg. It's actually a highly regulated sequence of molecular events that is controlled by some specific genes. And that's why in this presentation we'll delve into the mechanisms, that drive this gamete recognition and the binding, the eventual fusion, and the activation of what comes out of the fertilization, and that's the zygote. So, and the genome of the zygote, how that gets activated. So, whether you are here out of professional interest or sheer curiosity, I invite you to join me as we uncover the science behind the beginning of life for everyone. So, by way of introduction, fertilization is that process by which the sperm and the egg merge, forming a diploid zygote, and thus initiating a complex cascade of events that lead to embryonic development at the end of the day. So, this is a process that is very pivotal in sexual reproduction, and it marks the initiation of embryonic development. That's fertilization. And the precise genetic orchestration during this fertilization helps to ensure that there is successful fusion between both gametes to ensure that the genetic materials are united, are well merged, and thus laying the foundation for the development of a new organism. So, to give you further background into the anatomy of the sperm, so we can further appreciate the fertilization process, here is the sperm composed of the acrosome. The acrosome, this region, pardon me, this region is known as the head region, and you have the mid piece. Then from here downwards, you have the tail, while the very last part of the tail is the end piece. So within the head, you have the acrosome. That's the tip of it, the outermost portion at the tip. And acrosome, when you say somebody is acrophobic, that means the person has a fear for height. So that's like the peak of something. So that's the acrosome. And then you have the nucleus within the sperm. Don't forget the sperm is a cell too. So it has the nucleus in it. And then in the mid piece, you can see the mitochondria spread around. And what you have in the middle here is the axonema, the axoneme, and here is the centriole right here, which we'll see further here. So down here is so you have the terminal disc, which transits from it's the transition point between the mid piece into the tail region. Then you have the axial filament going there. So looking a little closer here, so within the head region, here is the cytoplasm of the sperm, and then you have the nucleus on both ends. By the way, when you look at the sperm in an anterior view in the front, in the frontal view or the anterior view, this is what it looks like. When you look from the side, laterally, it appears this way, so it's not all around. So right here, since this is the acrosome, so you see this yellow part, that's the actual acrosome. So it has a space deep to it, a small white space deep to it, and then another small white space outside it. So the acrosome itself is the yellow. The space deep to it is known as the subacrosomal space. That's labeled here, the subacrosomal space. Okay, that's the space that is just between the cytoplasm of the sperm and the membrane and the outer envelope. So then the space that is outside the acrosome is known as the periacrosomal space. So it refers to this small white space that is out there before then having the entire cell membrane, the outermost cell membrane. You have the nucleus, and then you have the vacuoles all around. Then the axoneme is usually found within the flagellum. That's the flagellar part, that extends between here down into the other region, and it plays a key role in the motility that is essential for fertilization. And that's why if there is a mutation in the gene that expresses the axonal protein, that can result into some motility disorders, that's including reduced sperm motility. So, and then you have the centriole around here. So it's usually a cylindrical cellular structure that is composed mainly of microtubules, and it's typically organized into triplets microtubules around there. So it plays a key role both in cell division and in the formation of the cilia and the flagella. So in this case, we appreciate the sperm as being characterized by its specialized structures that includes the head. It contains the nucleus and then the midpiece with the mitochondria around, and the tail for the mobility. And then the tail propels the sperm towards the egg, while the acrosome at the head here aids in penetrating the egg's protective layers during fertilization. And let me add that the distal centriole right here, so it gives rise to the axoneme in the sperm, so to ensure proper flagellar assembly and motility. So in fertilization, the sperm centriole also contributes to organizing zygous centrosome, so which is crucial for subsequent embryonic cell divisions. So now to the egg anatomy. So the egg is also a cell on its own, much bigger than the sperm. So the egg is a large cell with distinct features as well, including the zona pellucida. So the zona pellucida, you see this ring, there is a deeper green in there, and there is a lemon outer green, outer lemon green. So here the outer one is the zona pellucida. It's actually a thick glycoprotein rich layer that surrounds the oocyte. It's essential usually for protecting the oocyte and facilitating the binding of the sperm during fertilization. You see, but the vitelline, the perivitelline space, vitellus is the Latin word for egg. So remember, this is the egg, and this is the actual vitelline membrane of the egg. You see, so it's named vitelline membrane. That's the actual membrane that is surrounding the egg. That's the outer boundary of the egg. It's also the plasma membrane of the egg, because the cell we are talking about here is the egg. That's why we can call it the vitelline membrane. So it's the plasma membrane for the egg. So the space you find immediately outside that vitelline membrane is the perivitelline space. So, and that refers to this deeper green you have inside. So it's a narrow gap between the zona pellucida and the oocyte plasma membrane. And so in this structure, the zona pellucida forms an outer boundary. This is outer to the perivitelline space. And then you have outside the zona pellucida, you have the corona radiata, which is all around here. So each of these are the follicle cells that make up the corona radiata. So within the egg itself, you see the nucleus, which now has the nucleolus within it, and then the cytoplasm is the space that you find all other content in. Um, so you find the zona pellucida, of course, surrounding the egg, and then it has cortical granules, all of which are involved in preventing polyspermy, which we'll address shortly. And the egg serves as a nutrient-rich environment, and which provides essential components for the early stages of embryo development. So here we'll just appreciate the key differences between the egg and the sperm. So in fertilization, you have one egg released per menstrual cycle, while the sperm, of course, there are approximately 250 to 280 million sperm that are ejected during every ejaculation and are all aiming to fertilize the egg. In terms of size, the largest diameter cell in the human body is the egg, and it's approximately 0.12 millimeters. So, but unlike the egg, the sperm is approximately 50 micrometers long, and that is 10,000 times smaller than the egg. The sperm a single sperm is 10,000 times smaller than the egg. In terms of ploidy, the egg is haploid, but meiosis 2 is uncompleted until prior to ovulation. So remember, during the development, during gametogenesis, which we've also addressed its genetic basis, the egg is arrested at meiosis 2. During once a child, during the fetal life. So prenatally, the after the eggs are formed, they are arrested in the meiosis two. So the meiosis two is uncompleted. While the sperm is also haploid and it has its 23 chromosomes, and it's the maturation from the maturation of the secondary spermatocyte into the spermatid, which then undergoes spermiogenesis into the spermatozoa, will start occurring during from puberty. So furthermore, the mitochondria, you have between 100 and 200,000 in the egg, while the sperm, as understandable from its size, contains 75 to 100. Then other special features found in the egg includes the zona pellucida, which is that outer glycoprotein coat that you have seen, and the cortical granules, which which are enzyme packets within the cell. And then on the sperm side, it has a tail which enables it um, to propel, and then it contains mitochondria in the middle for energy, and then the acrosome, which is a packet of enzymes in the head. And then here is a graphical representation of what the egg looks like, and those major features, and then you see the sperm as well, showing those features that we discussed already. So there is acrosomal reaction of the spermatozoa is a prerequisite for an association between the egg and the sperm into what we refer to as fertilization. So, and this association, before you can have a spermatozoon and an egg, fuse together or fuse their plasma membranes, you need what is referred to as the acrosome reaction. So after a spermatozoon comes in contact with the egg, the acrosome, which is this prominence and the anterior tip of the spermatozoa, then undergoes a series of well-defined structural changes. And there is a structure within the acrosome known as the acrosomal vesicle. So what it does is it bursts. Okay, here is the acrosome. You have the sperm here. So the acrosomal vesicle bursts, and then the plasma membrane surrounding the spermatozoon fuses at the acrosomal tip with the membrane that is, you know, with the membrane that is surrounding the acrosomal vesicle in order to form an opening. So as this opening occurs, as this opening is formed, then you have the acrosomal granule, which is enclosed within the acrosomal vesicle, disappearing. So once that happens and the vesicle disappears, then there is dissolution of the granule, which then releases a substance that is called lysine, which then breaks down the egg's vitelline coat, thereby allowing the passage of the sperm into the egg. Okay. So once the sperm enters the egg, the acrosomal membrane region opposite the opening then adheres to the nuclear envelope, which is what happens here. So once it adheres to the nuclear envelope of the spermatozoon, it forms a shallow outpocketing, which then rapidly, elongates into a thin tube, and then the acrosomal tubule that extends to the egg surface, you know, and then fuses with the egg, the plasma membrane of the egg, which is happening here. So once this fusion happens, the tubule that has now been formed is what has established the continuity between the egg and the spermatozoon, and then this provides a way for the spermatozoal nucleus to then reach the interior of the egg. So and the whole spermatozoa injected into the unfertilized egg cannot really elicit the activation reaction or merge within the nucleus at the beginning. So that's why as the spermatozoal nucleus is drawn within the egg, then the spermatozoal plasma membrane breaks down at the end of the process, and then the continuity of the egg and the plasma membrane is then reestablished. So here are the cortical granules that you have. Remember, these we mentioned. So throughout this process, the sperm will come in contact with a number of protein receptors to make this happen. Here is the egg plasma membrane, which we refer to as well as the vitelline membrane. And then you have the perivitelline space, which is the space outside here. So here again are the cortical granules. So the sperm nucleus is here, and then the vitelline layer, which we just mentioned. So this is typically rounding up, summarizing what happens in the acrosomal reaction. And this acrosomal process is very important, for fertilization to occur. So in summary, you have the sperm making contact with the egg, and then next you have the acrosome reacting with the zona pellucida, and thereafter, and then releasing these hydrolytic enzymes. Then thereafter, you have the acrosome reacting with the perivitelline space. You have the plasma membrane of the sperm fusing with the egg. You have the sperm nucleus entering the egg, and then you have cortical granules fusing with the egg plasma membrane to render the vitelline layer impenetrable to. So to proceed, the here is a further display of the sperm, the acrosome nucleus, middle piece, and the tail of the sperm. And then you have over here, we have seen the zona pellucida, and then the polar body. Remember, the polar body actually plays an important, though indirect, role in fertilization. It's actually a byproduct of meiosis process in the oocyte formation, which we discussed during gametogenesis. During oogenesis, the primary oocyte undergoes meiosis 1 and then produces 2 cells. There is a large secondary oocyte, which will become the actual egg, and then there is a smaller polar body. So the polar body, you can consider it as a way for the body to discard the extra genetic material without wasting too much cytoplasm. And it's after meiosis 2, then the secondary oocytes get fertilized, and then it divides again, producing another polar body, and then the fertilized egg, which will become the actual zygote at the end of the day. So after that meiosis 2, if the secondary oocyte is fertilized, it divides again, like we mentioned. So the polar bodies don't directly participate in fertilization. They only ensure that the egg cell retains the appropriate complement of genetic materials. It's so that the egg retains majority of the cytoplasm and organelles, which are crucial for the early stage of the development. And then the polar body will essentially get discarded. It's more like a discarded cell, because the focus of oogenesis is to preserve the egg with sufficient cytoplasm resources. Yeah, so polar body also contains chromosomes anyways. It contains a haploid set of chromosomes just like the egg, but it doesn't contribute to the embryo. It only ensures that the egg has the correct number. So it's a way to help the body discard those extra materials. So yeah, so it's just a byproduct of the process that helps maintain the integrity and functionality of the cell. So we've seen the meiotic spindle being formed by the centrioles, and then the perivitelline space, and then the oolemma. Oolemma is also the vitelline membrane or the plasma membrane. Oo here is also referring to the oocyte or egg. So you see the acrosomal reaction, and then the zona pellucida penetration by the sperm. You see the sperm egg binding, and then you see the fusion of the pronuclei, and then the polyspermy block. So the key steps in fertilization includes the recognition, and then the binding, which has already included some of the acrosomal reaction, so penetration of the barriers, and then the fusion of genetic materials. So let's dive in now into the genetics of the of of fertilization. So here you have the father and the mother, right? And then you have the sperm coming from the male, you have the egg coming from the female, coming together to give the zygote that would become the child. So the sperm and egg carry each of them carries a unique set of genetic information which contains the instruction that will shape the characteristics of the offspring. Um, and the successful combination of both material from both parents during fertilization is what sets the stage for the development of a genetically diverse individual. Now let's get into it. Fertilization begins with the sperm recognizing and binding, like we discussed. The sperm recognizing and binding to the egg. So the ZP3, which stands for zonal pellucidar glycoprotein 3, is what acts as a receptor. So it acts as a receptor to for the sperm attachment. So you have the sperm protein isomo-1, which stands for immunoglobulin. It's an immunoglobulin superfamily member 1. So it's a sperm protein that is essential for recognition, for the sperm recognizing the egg. It's located on the sperm actually, and it's named after a Japanese marriage shrine, Izumo Taisha. So don't be surprised that there is no Z in immunoglobulin superfamily member 1. Okay, so it's Izumo. It's named after, it's a Japanese word apparently. So here is an egg, and here is a sperm, right? So for that recognition to happen, so this place was magnified, and the magnification of it is what you are seeing here. So this green part remains here. So here is still the oocyte, and then this is the sperm. Okay, on the sperm you have the protein known as izumo, and then on the egg you have Juno. Juno, by the way, is follicle receptor 4, and Juno is also not an English word, and that's why it's written this way. So it's an egg receptor protein for sperm binding. It's also named after someone. So in this case, it's named after the Roman goddess of marriage and fertility. She was known as Juno. So Izumo binds to Juno. Okay, so the eggs Juno receptor um, is yeah, so they both bind, thereby initiating a species-specific adhesion. So if there is mutation in any of these genes, it's going to prevent fertilization. So you can see Izumo on the sperm and then Juno on the egg for the binding. By the way, I may need to clarify why both the genes and proteins are mentioned here. So we mentioned that genes are segments of DNA that carry instruction for making specific proteins, right? So the genes will code for the formation of those proteins. So the genes, usually in genetics, the genes are written in the formats such as this. So in capitals and then italicized. So they are italicized when referring to the gene specifically. But don't forget that these genes are still the blueprints that will guide cellular processes by producing proteins through transcription and translation. So proteins, on the other hand, are the functional molecules that are encoded by those genes. So they are synthesized from mRNA, that's a transcript of the gene. mRNA is a transcript of the gene. So they are synthesized from the mRNA, and then they form specific biological roles. They perform different of such roles within the cell. So the proteins are generally written in the same format as their corresponding genes. The only difference is that it's the genes that are italicized when writing them. So the genes are in italics, while the proteins are usually not italicized. So ZP3 here is for the protein. So ZP3 here is a ZP3 gene that is coding for the protein ZP3. So ZP3 protein is not going to be italicized. ZP3 gene is going to be italicized. So Izumo1 is the gene that codes for the Izumo protein, formation of the Izumo protein, which will not be italicized. Juno gene coding for the Juno protein. So both are usually listed in the context of biological functions. especially in context of gamete recognition like this. It's often the protein product of the gene that directly carries out the function of that gene. A gene will not just function itself; it's the protein it forms that carries out the work. So, Zumo gene produces the Zumo protein, which is located on the surface of the sperm, playing critical role in sperm egg recognition. Similarly, the ZP3 gene encodes the ZP3 protein, which is a glycoprotein that binds to the sperm. So that is clarified. I hope so. To avoid confusion, you need to know these apart: the gene and protein, the kind of interplay. So, furthermore, fusion of the sperm and egg membrane is a genetically controlled process as well. So, you have the sperm protein PLC zeta. This sign is zeta. So, PLC here is phospholipase C zeta. Phospholipase C zeta. So this one initiates calcium oscillation that then activates. So it initiates calcium oscillation. You see that here, and this ends up activating the egg. So CD9 is another. It stands for cluster of differentiation. So it's present on the egg membrane, as you can see here, and it facilitates the actual fusion of the egg. Is very critical for it. So genetic mutations in these proteins would end up hindering um, fertilization, and it will not be able to go forward. And then polyspermy prevention is also vital. Okay, it's vital to ensure that we have a viable zygote, because upon sperm entry, that's the entry of the lucky sperm, right? Then you have the cortical granules releasing enzymes that then harden the zona pellucida, so that no other sperm can come in. And to make this happen, you have genes like the ZP1, that's zona pellucida glycoprotein 1, and ZP2, regulating this process to ensure that only one sperm fertilizes the egg, and thus avoiding chromosomal abnormalities. So you find that happening here. So this is the ZP1, this is the ZP2. So remember, the ZP3 is the one that mediates the actual binding of the lucky sperm. So while ZP3 ensures the actual binding, you have ZP1 and 2 preventing polyspermy. So that way they ensure that only one sperm fertilizes the egg, and to avoid chromosomal abnormalities. So after fertilization, the male and female pronuclei will end up aligning and fusing. So this is where we now have KIFC, not KFC. KFC is Kentucky Fried Chicken, right? So here is KIFC, and it's kinesin, not Kentucky. Kinesin family member C1, which plays a very key role in pronuclear migration and spindle assembly, the assembly of the spindle. So this is what helps to restore the diploid chromosome number. And you have some epigenetic reprogramming helping to ensure there is proper gene expression in the resulting zygote. So here is a proposed model, though proposed since 2021. So it's a proposed model of KIFC1 mediated breast neoplastic progression. So it's been thought and had been mentioned that KIFC1 may even assist pre-neoplastic and pre-invasive cells to progress by facilitating their uh, bypass of tumor suppressive barriers to malignant transformation and immortalization. I got this from a publication which is listed here. So where they believe the kinesin family member C1 is a potential actionable biomarker of early stage breast tumorigenesis and progression of high risk lesions. I just thought to let you know how much of research is going in this area. So the zygotic genome is activated shortly after fertilization, and you have transcription factors like the OCT4, that stands for octamer binding transcription factor 4. Okay, so you find the OCT4 and then the SOX2, that is SRY. You already know the what's SRY. Yeah, so the SRY is the sex determining region Y, sex determining region Y, which actually initiates. It's located on Y chromosomes, and it initiates spermatogenesis in males. So the SRY box transcription factor 2 is what is called SOX, right? So SOX, you also have the NANOG, which is the homeobox protein NANOG. So you have all of these coming together to regulate the activation. These are, yeah, so they come together to initiate the activation and also regulate the activation of the embryonic genes. So here is an embryonic stem cell gene. So they come together to do this. So epigenetic changes such as DNA methylation and histone modification, which we discussed in gametogenesis, also guide this early development. So here is a figure from an interconnected autoregulatory circuit. So genetic quality control is also critical for successful fertilization, and that's where genes, DNA repair genes like the BRCA, BRCA, which stands for breast cancer 1, and BRCA2. So BRCA1, BRCA2, that's usually how it's pronounced. They help to maintain genomic integrity to ensure that there is stability in the genome while defective gametes get eliminated. So you have genetic screening helping to identify chromosomal abnormalities in order to be able to improve the chances of healthy embryonic development as well. The BRCA1 especially is implicated in DNA repair, regulation of transcription, chromatin structure regulation, ubiquitination cycle, and in the regulation of the cell cycle. So, to summarize, there are a lot of molecular events that are happening in fertilization. We have seen recognition and binding, where you have specific molecular interaction between specific proteins on the sperm and the egg surfaces to facilitate recognition and binding. You have acrosomal reaction, which involves the release of enzymes from the acrosome to allow for the sperm to penetrate the zona pellucida and reach into the egg. And then you have fusion. So pardon me, this should be 3. So you have fusion of genetic materials of the sperm and the egg to combine the paternal and maternal chromosomes to form a haploid, a diploid zygote, and then this should be number 4. Okay, so activation of zygotes. So where you have the activation of the zygotes to trigger a series of events that are essential for early embryonic development. So once this happens and a zygote is formed and then it starts to divide, so you have the 2-cell stage and the 4-cell stage, the 8-cell stage, and then the 16-cell stage up to the 32, from which case you can refer to it as a morula, and then you have space developing in between it within the morula. Then you have this part will be known as the blastocele, and then this is the inner cell mass, and the entire thing is a blastocyst. And the development continues like that. So I just thought to also emphasize that specific proteins on the surface of the sperm and egg help to mediate crucial interactions that are necessary for the successful fertilization. And these interactions help to ensure recognition and binding of compatible gametes. So we talked about the genes and that they code for these proteins. So I just thought to let you know that these are they are specially acknowledged. So when you talk about surface proteins, so here is a list of these gametes surface protein. So Izumo 1 and Juno, you know the genes are code for them. You see they are not italicized here, so we are talking about the proteins now. So Izumo to Juno, right? So we know that we understand the etymology of this now, where the name came from. So Izumo is that sperm protein that binds the egg to initiate the fusion, and then the receptor, the egg receptor, is the Juno. And then you also have CD9, so which is So that's cluster of differentiation, right? The cluster of differentiation 9. It's part of the tetraspermine family. So it's involved in cell adhesion and communication. It's very critical for gamete membrane fusion and egg receptivity. So you also have the SPACA 6 on the sperm. So the SPACA 6 stands for sperm acrosome associated. Sperm acrosome associated 6. Okay, so it's very critical for sperm egg fusion after the zona pellucida has been penetrated. So it helps to create an interaction with the egg proteins like the Juno that we talked about. So there is also the TMEM95 here. So, and TMEM stands for transmembrane protein 95, transmembrane protein 95. Mem is membrane, T is trans, so transmembrane protein 95 is very essential for sperm egg membrane fusion as well, interacting egg surface protein. So it's located on the egg as well. Yeah. Next is CD881. So CD8, you can rightly guess that that stands for cluster of differentiation as well. So that's cluster of differentiation 81. It's another member of the tetraspermine family, and it works with CD9, which we showed here, to create fusion permissive domains on the egg membrane. So we can also see here, okay, the FMP, yeah, so which is otherwise known as Fertilin. So Fertilin is just the name. So it's originally part of the Adam protein, and let's just mention that. So while FMP facilitates sperm adhesion to the egg through integrin interactions, the Adam family, so Adam is created from, say, a disintegrin and metalloprotease. A disintegrin and metalloprotease. A disintegrin and metalloprotease. So it's named for their dual functions in disintegrin activity and metalloprotease activity. So it includes Adam 1 and Adam 2, which are known as fertilin. Okay, so Adam 1 is known as fertilin alpha, while Adam 2 is known as fertilin beta. So we also have sperm 1 pH, and sperm stands for sperm adhesion molecule. Sperm adhesion molecule 1. So pH refers to its earlier designation as a hyaluronidase. So it used to be known as a hyaluronidase, and it helps the sperm to penetrate the cumulus matrix that surrounds the egg. Then our ZP proteins, that's the zona pellucida. So the zona pellucida proteins, ZP1, ZP2, ZP3, we are familiar with all. There's also ZP4. So all of them are glycoproteins that form the extracellular matrix that surround the egg. Okay, there's also ovastatin. Ovastatin, ova, that's derived from ova being meaning egg, and astasin. So astasin is a type of metalloprotease. So this protein is a cortical granule protein that is released after fertilization to prevent polyspermy by modifying the zonal pellucidar proteins. So there's also the CRISP family, which helps to regulate channels and sperm motility. So CRISP stands for cysteine-rich secretory proteins. Cysteine-rich secretory proteins. So it's named for their high cysteine content, and it helps to regulate ion channels and sperm motility, thereby contributing to sperm egg interaction. So it's at this point we are quite familiar with a number of these genetic disorders. We discussed some of them already during the gametogenesis, so kindly refer back to that lecture. So certain genetic conditions may impact the fertilization process and thereby influence the ability of the sperm and egg to successfully unite, and one of them is the asthenozoospermia. Asthenozoospermia. So we are familiar with the word sperm, and zoo is an animal Like, yeah, that's why we have the zoo. So it's an animal living, and it's used in biology to refer to spermatozoa in this context. So zoo, and it's also referred to motility. So asteno, in this case, steno means to be strong, strength, steno, right? And to be asthenic means to be weak. So A here is a negative word. It means without or lack of. Astenosospermia. I'm a student of languages anyway, so it's important for you to pay attention to some of this etymology to be able to relate to what some of these terms mean. So asteno or astenosospermia. So to be asthenic means to be weak. So what is being weak here? Zoo here, which refers to animal. The reason why it's called zoo is because it moves as well. So the movement here of the sperm. So combined meaning would mean that astenosospermia refers to a condition that is characterized by sperm with reduced or lack of motility. So, and that's due to the vigor, the strength or the vigor, and this can strongly hinder the ability of the sperm to reach the egg and penetrate, and that would create a lot of fertility issues. So, PCOS, we also discussed that, is an endocrine condition in females and may lead to a lot of symptoms, including irregular ovulation, the viability of the egg, and the chances of successful fertilization. There's also the incompatibility in the major histocompatibility complex. So this refers to a case where you have genetic differences in the major histocompatibility complex between partners, and this can lead to incompatibility during fertilization, and it also can affect interaction between sperm and egg. So there are epigenetic factors that are implicated in this as well, such as genomic imprinting, which we discussed during our lecture on gametogenesis. So this can contribute to the regulation of gene expression. So here's typically sperm and an egg, and so the sperm from the father and then the egg from the mother, right? And so different genes are epigenetically silenced in the egg and sperm. Remember, they are not affecting the DNA structure, so it's an epigenetic factor. So they kind of affect one of the ways we talked about the histone modification. We talked about DNA methylation as one of the mechanisms of this. So at the end of the day, in the sperm, all imprints will get erased and rewritten with the paternal pattern. All right, that's what is called imprinting. Is following the species specific pattern. Okay, even the genes that came from the mom gets the imprints gets erased and rewritten with the paternal pattern. Okay, in the sperm, while in the eggs, all imprints gets erased and rewritten with the maternal imprint, even the genes that came from the dad. So that's why genetic imprinting is able to influence the expression of specific genes that are inherited from either the mother or the father, and thereby playing a role in early development. So after fertilization occurs by day one, so by day two, you start having cleavage. By day three, you start having compaction, different cell stage, different cell stage, additional cell stage. By day four, you start having differentiation of the cells. Then by day 12, which is post-implantation, you already have the bilaminar jam disc forming. I'm sure you've gone through all these in your biology, and we have that in our biology lecture as well. And then by day 9, the cell mass is differentiating. So we have the hypoblast, we have the epiblasts, and then that continues by day 7 when the implantation has occurred. By the 12, you have the formation of the third germ layer, introduction of the mesoderm, okay, and that is gastrulation. And then you have the mesodermal spreading, and by day 23, you have the amniotic um, uh, you have the amniotic regions visible and different from the chorionic. And then a lot of other events proceed like that. So the zygote undergoes all these mitotic divisions that form the blastocyst with increasing cell number. And these divisions lay the groundwork for subsequent embryonic development. So in summary, to conclude, we understand that the genetic control of fertilization involves a complex interplay of genes and pathways, right? So from gamete formation down to genome activation, each of the steps you find in between is tightly regulated to ensure reproductive success. And it's always important to understand this mechanism in order to address fertility challenges that may occur and to be able to advance reproductive medicine. So this is another nice illustration of post-fertilization process around here. So, to conclude, fertilization, we understand, is governed by precise genetic control. It marks the union of genetic materials, ensuring the intricate process of embryonic development. And this understanding is crucial. And we also have ongoing research that explores the advanced technologies and good intervention to better understand and manipulate. And insights from this research may have implications on fertility treatments. These are some of the sources that I consulted in putting this together, and I encourage you to visit some of them to understand and learn more. And this point concludes our discussion today on the genetic control of fertilization. I hope you've had a nice time, and I hope together I still manage to sustain your attention in our examining the critical roles of genes and proteins, as well as molecular pathways that are involved in this extraordinary process. Thank you for being there. And if you found anything interesting in what we have discussed, I look forward to hearing your feedback on that. Tell me exactly what you were happy to hear or something that you have learned new in this lecture, and feel free to stay tuned for more content. And we have a deep dive into a number of other topics and to explore the fascinating world of developmental biology together. There's still more to come. So until next time, stay inquisitive and continue exploring the science that shapes our world. Bye for now.